Well, good afternoon. I'm Dr. Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. I want to welcome you all to our Fall 2025 seminar series, Working Towards Better Water Quality Through Regional Collaboration. And with this seminar, we'll have two speakers per session. One is going to be an INRC funded researcher, and the other is going to be one of their collaborators who is also working on the same type of problem within the Midwest or Midwest adjacent. Today, our speakers are Dr. Alison Robertson, professor in the Department of Plant Pathology, Entomology, and Microbiology, as well as Extension Field Crops Pathologist at Iowa State University. And our second presenter is Dr. Damon Smith. He is the Vaughan-Bascom Professor in Plant Pathology and Extension Specialist at the University of Wisconsin-Madison. And they're going to be talking about a national collaboration to develop disease prediction tools to facilitate improved disease management and to protect yield. So I just have a couple of quick housekeeping items. This session will be recorded and posted to the INRC website. And if you have questions online, you can type them into the chat. If you're in the room, we'll have a question and answer session. All right, the floor is all yours.
Thank you, Kay. Good afternoon, everyone here in the room. Thanks for coming. And to those folks online, thanks for joining us today. Thank you to the INRC for inviting me to give this presentation. And I want to say thank you to Damon for agreeing to be my partner in crime in this presentation and in this project as a whole. So although this collaboration that I'm going to talk about is not actually related much to water quality. Damon and I have been doing some work on cover crops anyway, and we've been thinking along the lines of where we've developed models in this system, thinking about taking them further down into cover crops, etc. So we're right there on the outside, but hopefully you will get some insight into this collaboration and what we did. So this collaboration, the big name is called the National Predictive Modeling Tool Initiative. And the website is there, agpmt.org. And this initiative arose out of a white paper that was presented to the National Corn Growers Association back in 2018. In 2017, we'd had a meeting with the National Corn Growers. They'd had a stakeholder meeting. And at that meeting, someone had presented a proposal to map where all the corn pathogens were in the United States. And I was at that meeting and a couple of my colleagues, and we kind of said, well, but we know where the pathogens are, right? And what we really need is we need some prediction tools to tell whether we're going to get disease or not. And so it was kind of that we were then invited to write this pre-proposal to the National Corn Growers Association. So we wrote the pre-proposal, submitted it to them, and then we never heard anything back. And it turned out that the guy who had been excited about the proposal had then left the National Corn Growers Association. And so it went nowhere, and things were kind of quiet for a while. But we had an advocate, I guess, in Pete Snyder, who's the director of the National Agricultural Genotyping Center. And he really believed in the project and really thought that this would be a great national collaboration to get started. And so he did a lot of work on the Hill in DC, trying to get the funding for us. And so in 2020, January 2020, he called me and he said, I got money. So the initiative was established by Congress as part of the agricultural appropriation. And it was modeled after the barley and wheat scab initiative, which has been going on for the last 25 years. And so we modeled this National Prediction Modeling Tool Initiative after that initiative. And so this initiative operates under the USDA ARS. We have to wait each year for the Farm Bill to come through to hear whether we have funding or not, which is great fun. And so we started this in 2020. And the idea was that the initiative is hopefully going to go for about 10 years, at least 10 years. What we started with was we started with 3 crops. So we started with corn and cotton and wheat. Here in Iowa, corn is king, right? And so Damon and I are involved in the corn side of things. So I can't see my number there. You can't see the number there either, I believe. Yeah, it's 16, I believe it's $16 billion. So $16 billion is lost annually to diseases of corn, wheat, and cotton. That's a lot of money that's being lost, right? And a lot of crops. And right now, when we need to be thinking about feeding and fueling the world, we can't be losing $16 billion worth of crops. So what kind of diseases are we talking about? If I will advance, I'm not going to advance now. Hold on. There we go. So if we think about corn, right, the diseases that we're thinking about is southern rust. For those of you who've been out in the cornfields this growing season, you will have seen southern rust everywhere on corn, right? So southern rust has been a big problem this year. Another disease that you've probably heard of a lot is tar spot. Tar spot was first reported in the US in 2015. And then the last 10 years, it's just expanded the area over which it is and has caused tremendous disease losses in some states and in some years. Then other important diseases, we have our gray leaf spot, northern corn leaf blight. They've been around for ages, but depending on the year, we can have outbreaks of those diseases and we can lose a lot of yield to that. And then we have the ear rots. The example that I have here is gibberella ear rot and stalk rot, and associated with the ear rots are mycotoxins. So the one thing that I want to draw your attention to with these diseases is that, yes, we see them every year, but the impact that they have on the crop varies from year to year, right? And so we don't know what's going to be the problem in this year. And so the idea behind the National Predictive Modeling Tool Initiative is to be able to develop these tools that farmers can use to predict whether they're going to have a problem with tar spot in their field this year, and therefore whether they need to apply a fungicide or not. So the goal of the overarching goal of the National Predictive Modeling Tool Initiative is to develop these research-based tools that will help forecast incidence of disease and mycotoxins affecting US row crops. So Damon and I are members of the Corn Research Area Committee, and then there's also a Cotton Research Area Committee and a Wheat Research Area Committee. And I chair the Corn Research Area Committee with Damon as the co-chair. And obviously the other research area committees have their chair and co-chair. And then we have an executive committee, which is half a dozen of us who will make decisions if need be. So when we started out in 2020, so this was the fun thing. In January, we got told I got the phone call, we had the money. And now I had to set up this research ready for the 2020 growing season. And the first thing that I needed to do was to identify different collaborators. And so I guess this map is kind of getting a little bit ahead of us, but you can see for 2020, there were 7 states that were involved, right? So showing those are the participants there. And then we had the cotton, obviously down in the southeast, so several states collaborating there. And then the wheat up in the Pacific Northwest, and then the plains, and then also down in Texas. So this is a national collaboration, and that was one thing that Pete, the guy who kind of spearheaded this whole initiative, it had to be a national initiative. That was the way that he'd sold it to Congress, right? That it was going to be this national initiative across as many states as possible. Also, part of the initiative, well, Los Alamos was involved in this. So Los Alamos is out in New Mexico. They do a lot of modeling of animal diseases, human diseases. And so the idea was that they would come in and help with the plant diseases and just assist in that way. We worked with also the USDA ARS Research Service. They were going to be involved. And so we would be working with scientists in USDA ARS. And then, of course, the National Agricultural Genotyping Center, which is what Pete directs. And we had a role for them, that they were going to play a role in the initiative as well. I just want to say that as we step forward into 2025, the initiative is growing. So we've added more states for corn. I don't believe that cotton or wheat have added any more states. I couldn't find that information. And then the other thing that we've started adding is the pulse crops. So up in the Dakotas. So this National Predictive Modeling Tool Initiative is for all field crops, right? And the idea is that hopefully we will get more money from Congress to be able to add more field crops to the initiative. And yeah, that's been a little bit difficult, but we have added the pulses, and they're kind of in this preliminary phase at the moment, and then hopefully will expand over more of the US. So, like I said, Damon and I are part of the Corn Research Area Committee, the chair and the co-chair. And so we have two goals as part of that committee. One of them is to develop and to validate these prediction tools for diseases and mycotoxins to help with management decisions, especially things like fungicide efficacy and, well, not fungicide efficacy, just fungicide decisions, right? Whether to spray or not. And in so doing, by managing these diseases of corn better, hopefully we can reduce yield loss, and that can also help farmers profit more, right? Because they're not using yield, but also they're not using fungicides when they don't need to use them, right? So it's an input that they're not going to pay for. And then our second goal, which is very important to us, and Damon's really going to discuss how we've been doing that, is enhanced communication and end user education and outreach for our audience. And our audience is farmers, anyone in ag business, the research community, grain processors. So anyone who's involved in corn, right? We need to get our tools out to this. So when we started the project, this one thing that I wanted to bring up is this was really exciting for us because it's exceedingly hard to get money to work on corn, right, from a federal granting agency. So a lot of the corn epidemiology work about diseases has not been done for a couple of decades just because there's no money. And so this was just a great way to get money to research that epidemiology and then to develop these tools and pass them on to the farmers and really get a handle. And it was maybe serendipitous as well that we had TaskSpot, right, coming in. We didn't understand that disease, and so there was a lot of work that had to go on to TaskSpot to understand that epidemiology. Okay, so building the team. How did we go about that? So when I got the phone call in January, I was told that it had to be national. So I had two colleagues, one from Kentucky and one from Missouri, who had been with me at the National Corn Growers meeting. So I reached out to them and said, hey, we got money, will you join us? And then none of us model plant disease. So obviously I needed a modeler. And so that's where Damon and then also a colleague at the Ohio State University, they both model diseases. Damon's had developed an app for white mold on soybeans. And so they were the perfect choice, right? So now we have Wisconsin, Ohio, Iowa, Missouri, and Kentucky. And then we had a phone call and I kind of bashed some ideas around about who else we needed, because we needed some Southern colleagues to help in this. And the one thing that's really neat about working in field crop pathology, which I think is very unique compared to anything else in the US, is that we're very, very collaborative. We do a lot of research together. We do a lot of multi-state research trials, etc., together. And we've been doing that since 2005, when fungicides started to be used on corn and soybeans across the state. In order to get good data to be able to share with our growers, we started to work together and share our fungicide trial data. And then as questions came up, someone would say, hey, I want to look at this. Anyone else interested? And then we would have these collaborations across the states. And so that's what's really, really neat about this group of individuals. So expertise was important, right? I kind of covered that already. Because this was working with epidemiology of diseases, it made more sense to get field-based corn pathologists, because we would be out in the field, right, collecting data there. And then having the predictive model experience was obviously really important, and that's where Damon and Pierce came in. And then I kind of already mentioned this as well, but we have a history of collaboration. We work together a lot. We're always working together at our national meetings that we have every year. We're always planning research trials that we can do across several states. So it wasn't really, we're used to working with each other. One of the things that we also wanted to think about was that everyone had to be team players. And then we also needed people who were trustworthy. I was thinking about this. We didn't want anyone who's a prima donna, right? We're all contributing to the team. This is a team effort. We have to be, we're sharing data with someone, right? All that data goes to Damon's lab, where he then develops the models and everything. And we trust Damon not to, pull the rug out from under our feet, right? He's there. So we also had to have a shared vision. So one of the things that we've had since the fungicides have come into maize and soybean production is that just having responsible use of those fungicides on those crops. And so that was our shared vision, is that all of us want fungicides to be used responsibly, and here was a way for us to develop tools that would help growers to be more responsible with those inputs. So to establish our collaborative culture, while we were planning, when we got the news in January and we got the team together, we then had meetings to define our goals and objectives. We spent a lot of time talking with each other, discussing what we wanted, what did we need to do, what should we be looking at. And it was very important that everyone was allowed to share what they thought that we should do, and that everyone's expertise was valued. Ideas were valued. Some ideas got tabled for another time, but we worked together really closely on defining what we wanted to do. And Damon's going to take over in a couple of minutes and just explain what we did. We still have monthly meetings, and we still reevaluate our goals and objectives annually to make sure we're on the right track, make sure we're developing models for the most important diseases coming up. What do we need to develop a model for? How do we need to refine a model? Is there anything that people are not happy with? So we meet monthly to do that. Okay, Damon, I'm going to pass it to you now, I think.
Sure. Yeah. Everybody hear me okay in the room, Allison?
Um,
I think we need to turn your volume up.
we're going to turn you up.
Okay. How about now?
Just wait.
Okay.
Looking for the right. Oh, here we go.
Okay, can you say something again?
Sure. Yeah. Sound better?
Yeah.
Okay, awesome.
Okay. Yeah. Yeah. Thanks, Allison, for the invite here. I'm happy to talk about this multi-state collaboration. It's been a really interesting and rewarding experience. And I will say I think what we really learned, and it was on that last slide, is that really having that trust is key here. And I think, if anybody's looking at trying to get multi-state projects off the ground, or you're in the middle of one and you're just trying to figure out how to make it go even better, that really is key. And I think we learned that through this whole process and how important that is. If anybody didn't trust each other, I don't think we'd be standing here. And so that was really important for the group. And as Allison points out, there is a lot of load on each of us. And, for me, I worry every day because all the data is coming to our lab, and folks trust that I'm going to do the right things with that data. I'm not going to scoop them on things. And it's been a really good collaborative environment, but there certainly is a lot placed on a lot of us in this project. So it's been great. I'm really love this project. I'm looking forward to it continuing on. Fingers crossed that the funding remains. We'll see how Congress approaches that. So when we came on in 2020, one our initial meetings really were focused around, all right, how do we generate data? Because we there's a lot in the textbooks about where these diseases come from, how they move. But interestingly, when you really started to dig in the literature, there weren't a lot of fine details published about some of the nuances about the disease cycle and spore flight and some of these really important pieces in the epidemiology side of things. And as Alison pointed out, I'm trained as an epidemiologist, so I do quite a bit of modeling work on plant diseases. Have done so ever since I was a master's student at North Carolina State. And so Pierce and I sort of sat down and said, well, what pieces in these specific cycles for diseases do we not understand? And, really a lot of it came down to when spores fly and when those spores land, how long does it take to actually get disease formation? We call that incubation period. These are really important pieces from a management standpoint. That helps really allow us to get that fungicide timing really optimized. And so we felt like the best way to get at some of those fine details was to do these coordinated small plot trials. So we set up a protocol, and we'll show you that protocol here in a second, to do some really finite work to really try to understand the biology around these organisms and apply that to the epidemiological understanding that we had up to that point. The other piece of this, which was really interesting, is to actually go out in commercial fields and then take disease data and then use that data to both validate and also augment what we were seeing in the coordinated small plots. And this was very different from what folks had done in the past in this realm, because it's sort of just considered that commercial fields are hard to integrate into modeling work, and that you don't have replications, and so you can't really understand the variability and all these sorts of things. But the approach we took was to have a lot of commercial fields. And I think at the end of the day, we actually had more commercial field sites than we actually had small plot trial research sites. And we were actually able to combine that data, and I'll talk about that here in a little bit, together in both training and validation data sets to help with our modeling work. So Allison, if you could click to the next slide there. This is just a sort of a diagram of what the first small plots look like. We've since had derivations of this, but this sort of got us off the ground. And there were three primary diseases at the time in 2020 that we were really interested in. It was tar spot, northern corn leaf blight, and gray leaf spot. And so, depending on the primary disease of focus in a particular location, we didn't really dictate that. We let folks sort of decide, all right, I want to focus on tar spot in Wisconsin. And, the folks in the South wanted northern corn leaf blight because they have early northern epidemics there on their early planted corn there. And then some in the mid-South wanted to look at gray leaf spots. So they chose a resistant susceptible hybrid that they knew there. What we want to do in these epidemiological studies is try to get a lot of variation in the plot so we can then use models to try to explain that variability. So you have to have a training data set that has a large amount of variation. The best way to do that in plant pathology is with hybrids and hybrid resistance. So we had two hybrids there, and then we also wanted to manipulate inoculum load. So with most of these, actually all three of those particular diseases, they're all inoculum based in terms of the initiation of the epidemic. But what wasn't really understood is how does inoculum potential, if you will, or how much inoculum is there, how does that dictate when and how severe the initiation of that epidemic might be? It's sort of stated in the basic plant pathology books, but when it came down to like really trying to find out the details, there wasn't a lot in the literature about those details. So we really wanted to do some work there, and so we also had you could either manipulate your inoculum load through tillage practices. So for example, in Wisconsin, we had no-till sites for corn-on-corn no-till for high residue, and then we had a tilled following soybean site for low residue. Some other locations, for example, in the South, where they have a lot of residue turnover, so it's hard for them to actually keep a lot of residue there. They did planting date studies, so they would plant early and then have another one late. The idea there is certain diseases, the inoculum load will actually be higher later in the season. We do know that. And so through those different designs, we were able to sort of manipulate the pathogen side, if you will, if you're thinking about the triangle. I know Alison always likes to make the finger triangle, and that's a really important piece there. And that's really what we're trying to figure out is when is the pathogen present? Do we have that susceptible hybrid? And then the weather that really comes down to drive that. The other piece here was spore traps. Yeah, and so what we also wanted to do is try to understand spore loads at various times through the season. And we actually deployed two different types of traps. There's a sort of a standard in plant pathology. There's a what's called an active suction trap. It's we know it as the Burkhardt spore trap. Been a lot of publications around that, but there's also some cheaper traps out there which are sort of passive or semi-passive. And we chose to use a semi-passive trap, which entailed a grease slide that spun on a garden motor. And I'll actually have some pictures of that a little later on in the presentation. And so, next slide, Allison. What we then did is trapped on a weekly basis. So we would deploy our traps at the beginning of the week, and then the following week on that same day, we would come back and change the traps out. And then we were interested in using quantitative polymerase chain reaction or qPCR technology. So this is newer technology. Traditionally, folks, when they obtained a spore trap, you would look at that spore trap under a microscope and actually identify all the spores on there by morphological keys. And as you can imagine, that's very time and labor intensive. And there's better ways to do that now with the technology we have with gene-based technologies. So we were very interested in generating qPCR data, which is also quantitative. And so we do have now qPCR data for all three of those primary pathogens that cause those diseases. And we were also interested in the inoculum load at the beginning of the season. So we also collected corn residue out of those various plots and then sent that up to the NAGC. And then actually initially during the pandemic, Alison and my lab actually did a lot of that work on the qPCR side. But now that a lot of that work has moved over to NAGC as funding is moved around a little bit and relieved some of the load on our labs because qPCR technology, while very sensitive, is quite expensive. And so it's helped a lot being able to pair up with NAGC to run that and generate all this data. So next slide, Allison. We also, at the same time, we were trying to get an understanding of what was going on with actual pathogens out in the landscape. We also were taking very intensive disease data so that we could then relate that back to spore flights. Remember, we're very interested in things like that incubation period, the time it takes from a spore to land on the leaf to when we actually have disease develop. So we were actually tracking at three different levels. The kind of a low level or ear -1, mid level at the ear leaf, and then above the ear leaf, we were taking ratings. We had a minimum of at least 3 ratings per season that we were trying to get after, and we were using rating diagrams that are digitally generated, and you can see some examples there for gray leaf spot right in the center of your screen. So actually showing you what those real percentages are. And then we were looking at 10 plants per plot. So somebody had to go out into those plots, take those ratings. If you had multiple diseases, it was multiple ratings. So we had undergrads and postdocs and graduate students all working on this, spending hours and hours intensively rating. But again, able to really generate high detail data that we're going to be able to actually mine for years. So that's the other nice thing about these types of multi-state trials is that you generate a data set that if you curate it correctly, can actually be available to mine for many different questions that could be asked for many years. And I see us, at least a decade from now, still able to mine the data that we generated even in the first iteration of this project. Next slide, Allison. So just to sort of give you an idea about how this goes, and I won't bore you with all the math that goes behind this. That's a whole different talk. But this is just sort of a flow diagram, and that QR code will actually take you to a paper about our tar spot model that was developed based on the data set that we generated. And you can kind of see the process there if you really want to geek out and try to understand what was going on, sort of on the back end of things. But very, very broadly, what we were doing is to initially, we wanted to get a disease model together. We're currently working on the spore model. So, sort of systematically, you have to separate those things. They're sort of two different things, and different weather parameters can actually affect those two different processes. So the first thing we were after was generating the disease data. So what weather parameters really drove the increase in tar spot, for example. And so what we did is we took all the disease data that we had collected, and then we blended that together with a weather data set. And over time, I've become very interested in high quality cloud-based weather data. And Alison mentioned that I had done some work on some smartphone apps in the past. Sporecaster was the first one. We actually generated tar spotter, which came out of this as well. And in those tools, you really have to have a remotely accessible form of weather. And so, after a lot of work, and that again is another whole talk, we actually landed on the IBM Environmental Intelligence Suite. So, the IBM data set, that's a 2.5 kilometer gridded data set, coarsely generated off the HER, so the high resolution rapid refresh NOAA models. They do some data smoothing and some proprietary things to that data, but it's really quite high quality data. And we've run a lot of correlation and concordance analyses against in-field weather station data. So we know it's a high quality data set. We then take that weather information that comes in from that cloud. We have the GPS coordinates for each of the plots or the commercial fields. So remember, we had both of those types of data actually going into the disease data collection database here. We blended those together, making sure everything was aligned based on GPS coordinates, and then we were able to do some initial correlation analyses to really understand what we call window panes. And so, again, sort of asking the question is how long does it take? What weather really impacts that disease development after that spore lands there? And that's how you sort of think about a window pane. And depending on the disease, that can be a different type of time, right? It could be 7 days for one disease, it could be 14 for another, 21, maybe even as long as 30. And so what we can do very rapidly with our statistical software, things like R and SAS and Python, is be able to actually test different window pane lengths. So we did a lot of that work initially looking at those correlations. Once we landed on some really important variables and various window panes, we then could actually test those. And we were very interested going into this. We had done some work with sort of simple machine learning, so logistic regression models. And then we were also interested in more deep learning type models, so random forest and artificial neural networks. And again, you can find a lot of this in that paper at that QR code. And we tested all these various models together. At the end of the day, we actually landed on two different models for tar spot. We actually run those together at the same time, and then generate basically an output. So up to this point, we've done a lot of the technical work, and then it comes down to, all right, how do you actually get sort of the technical side of things out to the farmer, right? And so we have to develop a decision support system and actually get that tool deployed. So at the end of the day, that's really the important piece because that's what the public actually sees. And after about 5 years of heavy lifting and various iterations of things, we've gotten to, Allison, if you could go to the next slide. We've gotten to this dashboard, which is actually shared on the Crop Protection Network. I'm sure a lot of folks at ISU are familiar with the Crop Protection Network. Darren Mueller is one of the directors there. We like the Crop Protection Network for this type of tool because again, it's a collaborative effort across many states. And so we thought it was just a natural fit. We did originally have the smartphone apps. Those have since gone away. We learned a lot out of those tools. We learned how to make decision support systems better. While we're still, tweaking these tools even today, this QR code will actually take you to the dashboard site. You'll actually find various models for various crops there, but you can see Allison ran some tar spot forecasts here for early September across Iowa. And so just in a snapshot, folks can access this in a few seconds. They can get those risks indices. We actually run a 7-day forecast. So you'll see there's a line there on some of those outputs along the right-hand side of the screen. And there's a dashed line which depicts today. And then the 7 days ahead is actually a true forecast. So we're actually pulling in forecast weather data to try to get a forecast, which is really important for farmers trying to make a decision, especially potentially with rain and that sort of stuff coming in. So we have a really nice tool, and I'll show you some impact here in a second. Allison, you can go to the next slide here. We've had a lot of accomplishments. Again, Allison pointed out we went from 7 or 8 states originally to now we have 11 states involved in just the corn rack. We have a lot of site years of data, more than 100 site years of tar spot, gray leaf spot, northern corn leaf blight. We've now added southern rust to that data set, and I think after this year, that'll probably be a higher prioritized model for development. We also have an ear rot model that runs actually publicly now on the front end of things, and so we're always trying to grow this. We're doing a lot of outreach around these tools as well. So each of the colleagues that are part of the MPMTI, we generate a slide deck each year, and we send out the impact data, and they talk about it during winter meetings and field seasons and all that sort of stuff. We've published peer-reviewed papers. We've generated primer sets. We're working with the USDA to dig in on mycotoxin tools and all those sorts of things. So there's been quite a few accomplishments there. As far as the tool specifically, Alison, if you could go to that next slide deck, it's been really interesting to watch how these tools grow over time. And we went from, with the standalone apps, things like Tar Spotter, we went from maybe 1,200 users per season and maybe about 10,000 repeat page views to this past season. I had our data scientist pull up some numbers the other day. This just floored me. I hadn't seen these numbers until just the other day. We had 4,100 standalone users with the CPN tool this year alone, and over 31,000 page views, which is phenomenal. So almost 3 times what we saw with our smartphone apps, and I think a lot of that has to do with the fact that we've now housed all the different tools in one place. We're pushing multiple crops. So we also have soybean on there, as well as some other crops that are sort of there's data being generated in other areas and other initiatives and those sorts of things. And CPN's been a good clearinghouse for a lot of that. Just so you can wrap your head around what that means financially on the farm. On average, a tar spot fungicide, for example, would be around $30 per acre, give or take. Some are more, some are less. It also depends a little bit on your aerial application costs. But if you can get that timing dialed in and you decide you don't actually need a fungicide, that's a big savings right there. But you can also optimize that application timing, and we've put some numbers to that. It's somewhere in the $1 to $10 per acre range if you can really get that optimized in terms of return on investment on the farm. So, not only is it just the savings, but in years, like this year where we've had high disease levels, being able to really well time those fungicides gives us a lot of return on investment in those products as well. Not only do we want to try to, keep them around a long time, we want to try to, help farmers use them judiciously there as well. We've also generated other outreach things that are on the crop protection network. So we have a sort of a layperson or public facing page about how the tar spot tool actually works. We also generate outreach tools around just trying to optimize the timing based on these tools and that sort of thing. And you can kind of see the impact that we had in 2024 there. So just to finish up as we kind of wind down on time here, I do want to leave some time for any questions for folks. Our successes, I think Alison and I agree, we had 3 additional states here, which is huge, being able to sort of reallocate funding and add more states and get more collaborators. And, more people is more fun and generates more data. We now have 3 predictive models running. We have a really collaborative culture. I mean, we're probably the tightest that I've ever seen a set of researchers in any of the worlds that I've worked in, and I've worked in quite a few different worlds between my time at NC State. I was also at Oklahoma State for a while. I mean, I'm just really impressed by this group and what they've done. And now we've teamed up with the Crop Protection Network to be even more collaborative across the nation and being able to really push those tools out freely to the public. And I promised there was an image of what the spore traps and weather stations and all that cool equipment. This is actually a shot of some plots that were here in Wisconsin. That big thing that looks like it's going to take off right in the front there. That's actually a Burkhardt spore sampler propped up at 6 feet or approximately 6 feet, where it's sampling air. The middle device there, that's of course a Campbell Scientific Weather Station there. And then way in the far back, you might be able to make it out. There's a shepherd hook with one of our low-cost semi-passive samplers. So again, each of the states had setups similar to this, maybe not with a weather station in every state, but they definitely had these traps that we could get quite a bit of data off of. Next slide, Allison. I think the challenges here, and this is I like how Copilot put wheat heads on top of corn. I was dabbling around with AI the other day and just got a kick out of this, so I decided to put it in here. But one of the big ones, and going to be an ongoing problem, I think any hurdle or any multi-state type of collaboration is going to run into this funding. Funding is always the big one. We've seen just flat funding basically the whole time, and we've tried to add folks, and so somebody has to take a cut in pie. And Alison and I have taken cuts over time here, as well as others, not just us, but others have taken cuts as well to try to bring others in. And so you really, at the end of the day, you have to sort of set your ego aside for multiple reasons when you come into these types of collaborations, because you got to be able to generate an inclusive environment where folks are willing and they trust you to get these things done. But you also have to set aside that you're going to land a really big grant, like generate a lot of funding and that sort of thing. That doesn't always happen. You're often doing a lot of work for not a lot of funding, and that's just sort of the way it is. But we are funded and we do get some support there, which is really good. One of the other big things has been data capture and curation, and that's been really a pinch point at this point now. And I actually have a data scientist here at Wisconsin helping us try to generate an automated database where we can pull in all the data we are generating and then get that stored in a safe place, and then also make it accessible for folks who want to do different things. So having the right metadata in there and being able to have it a searchable database that folks can access over the web. And up until this point, we've been saving things in large Excel sheets, which is not the way to do it. We don't want to be doing that kind of stuff anymore. And then you would send that big Excel sheet to somebody, and then they get overwhelmed with the data. If we're going to really help folks be empowered for model development, they need to have access there. And then the final piece, and one of the big things we've gone away from was really app development. We were doing a lot of stuff on standalone apps and tools, and some of while those are kind of cool to be able to like pull up a standalone app on your smartphone, there's a few things there that really are big bottlenecks, and one of them is programmers. Programmers are not trained on both platforms, both the Android and Apple platform, and so you actually have to hire two programmers if you're going to make an app. And I've been them through this whole thing multiple times, and it really gets expensive. The other thing is those apps constantly have to be updated, re-uploaded to the stores. So there's a lot of curation and those sorts of things, and it's quite expensive. The other thing that's happening, and you can see it with what Apple is actually doing, they just had their big Apple event yesterday, but their smartphone operating system is actually converging with their desktop operating system, and probably within the next 5 years, we're actually going to see one common operating system across all platforms, and most things are going to be running via over the web. And so we're trying to get ahead of that a little bit with the CPN tool to get it into one location. Run it over the web so that it's sort of agnostic in terms of the operating system that it can be accessed on. And so those are really the main challenges that we run into. Alison, I don't know if you have anything else to add there, but those are kind of the bottlenecks that we've run into. But I'd say overall, it's been a really rewarding project, and I really enjoyed the time, and I love working with Alison. She just tells me how it is, and I like that. I'm a very to the point person, so it's been a great working relationship with Alison. So with that, I think we have a thank you slide, Alison, and maybe some time for questions.
Thanks, Damon. Any questions? What questions? Yes.
Hi, as Damon, this is a great talk. So, I'm very interested in the model prediction of the disease. So I saw the model is kind of heavily reliant on the climate, those data. So I'm a soil scientist. I often think about question from the soil property perspective. So I'm wondering how much of those data parameters can expand the disease predictions. And if addition of any soil property to your model, whether if you think it can greatly improve your model prediction of the disease or not. Thank you.
Damon, did you hear that?
I did. I could hear bits and pieces, but I didn't really get the whole thing. Could you just repeat it for me, Alison?
Yes, so it's about the data prediction. We're relying on weather data, but we have a soil scientist in the room, and so she was wondering if we're going to look at other factors like soils that might impact diseases.
Yeah.
Yes.
Yeah. That's a great question. And again, I've been doing disease prediction modeling for more than 20 years now. Started doing this as a master's student. And at that time, we were very interested in a lot of the soil parameters. The trade-off, though, is that we can get things like soil texture, soil moisture, soil temperature, all these sorts of things by going out, sampling, and then putting a weather station with the correct probes out, right, to track those sorts of things. And that works well if you have a hardwired weather station. In the field crop side of things, farmers are they're more adverse to actually having an on-site weather station. They'll tell you they want a weather station, but they do not want a weather station plunked in the middle of their field. Fruit growers, and I did some tree nut work when I was in Oklahoma. They're a little more willing to have a hardwired weather station out there, but then you got to babysit it. And so over time, this is why we wanted to gravitate also to some of the cloud-based data. The trade-off, though, with cloud-based data is then with soil parameters, we have less accuracy there. And there's some nice papers that NOAA's put some papers out, especially looking at the HRRR weather data sets or the high-resolution rapid refresh weather data set. They've shown that when you're looking at the atmospheric or low atmospheric weather variables, They have very good predictive ability, but when they go to like things like soil moisture and soil temperature, we lose accuracy pretty quickly. Although the HER model is actually probably one of the best out there right now. I do have another data scientist right now. He's working on developing a new API, a weather API for us, and that is one of the big things I've asked him to do is to start to pull in some of those soil moisture and soil temperature data in here because, as you point out, having those data here will only improve our training ability for these models. But it has been admittedly difficult up to this point to have that cloud-based weather data and not be able to include some of those soil parameters, which are very important.
Thanks, Damon. What other questions? What didn't we talk about that maybe?
On the running of the project, great talk, thank you. What would you see as one of your biggest problems on the actual running of a huge project like this with multi-state nationally?
What are some of the problems associated with a big collaboration like that?
And then follow up on that is also choosing your leadership. How did that?
Yeah, I don't know how the leadership got chosen, but I think from just from working on other collaborative teams, and I don't think that we had this problem, but everything we did was standardized and everyone followed the protocol. I've worked on other big collaborations where people don't follow the protocol, and then when you get the data in, it's not the data that you asked. You know, Damon talked about how we collected data from 5 leaves, 10 plants, at least 3 times during the growing season, right? If someone had just collected data from 10 plants from 1 leaf once during the growing season, that just because that's what they wanted to do, it wouldn't have given us all that information that we needed, right? So it's not a problem that we had, but I see that as a problem with like collaborating with other people, is that if there's a protocol, it needs to be followed, right? So do you have any thoughts, Damon?
Yeah, I mean, so Alison and I are both part of another large collaboration which involves uniform fungicide trials, and that's exactly what happens. We have less we sort of have less I don't know how to put it, but buy-in, I guess, in terms of the protocol itself. So we'll generate the protocol, but then folks sort of just decide that they're going to do their own sort of thing sometimes, right? And so you get these data sets back and they're just sort of junk. And unfortunately, you have to throw them out. And I think where the NPMTI is a little different is we're meeting monthly, right? And so we're sort of talking through this. And I think that's one of the big hurdles is like you're gonna sort of spend more time on the logistics sides of things, right? Like having meetings, talking things through, going through multiple iterations of a protocol before you sort of arrive at what that looks like. But the trade-off there is that everybody had buy-in on that protocol. They had some say in that protocol, and what that does fundamentally is that it builds trust, right? So at the end of the day, that sort of vulnerable trust that we have with each other is really what drives a lot of these projects and really drives our universities, right? So if you don't have trust in your administration, right? Like we know how that goes, right? And so I think that's really the key piece here. And so you have to spend that time having those multiple meetings because really that's what really gets folks motivated and it helps them have that buy-in.
So your team is mostly pathologists, but as you look at the topic like water quality, where there's a lot more people involved, what would you do differently given the diversity need to be or want to be around the table at one meeting?
So did you hear that question, Damon? So like if we think in terms of water quality, there's people from so many different disciplines, and what might we do to form a team there? I mean, for me, Dan, I've been here for 20 years. Damon's been at Wisconsin for, I don't know, 15 years or something. So I know a lot of people, right? And I know who I trust and who I can reach out to, who can help me, right, on a project. And so those would be the first people that I would go to. And then I might say to them, hey, we need someone with expertise in manure management, right? Do you know someone who you think would be a good fit, and who's a good team player, who we can trust? And then, so that's the way I would go about it. But yeah, I mean, having been here for 20 years, I know people who I would work with again. I also know that, I mean, there are people out on campus that I would not work with again. So, yeah. Damon, you have any comments?
No, I like that. I'm actually part of some bigger projects here at UW. Well, not bigger, but more multidisciplinary type projects. And that's exactly how we sort of handle it here, although it's not quite as smooth. Admittedly, dealing with a bunch of plant pathologists of the same discipline, that it admittedly is an easier hurdle, right? But we do some IPM things, so we have entomologists and weed scientists and all that sort of thing, and related to some cover crop work that we're doing. And really that's key, is like you just have to surround yourself with sort of the folks that you trust, and then empower them to be the leaders to make those connections in their disciplines. I think that's, so the web gets a little more complicated there, but again, if you empower those folks that are sort of leaders in those particular areas, I think you'll find that like you can get those folks sort of motivated behind that, right? Because folks just want to be surrounded by folks that think the same that they do, right? So entomologists want to be surrounded by an entomologist, and plant pathologists are the same way, and so are weed scientists when we look at the pest management side. So I think that really is key, is you just get those key players involved that you trust, and then that sort of motivates the rest.
Not to push back too much, but for example, on the water quality side, isn't there a chance that doing that, we isolate ourselves to be productionists when you work at an agricultural college, and it's harder to facilitate that conversation to people in the city of Des Moines that have some say in water quality as well?
So if you were on my team, Dan, I would say, that's a great idea, Dan. Do you know anyone in Des Moines who we should have to join us on the team? And I think that's one of the important things about, if you're going to establish these collaborations, is having those initial meetings, right? And having people play the devil advocate, right? And say, well, what about the folks in Des Moines, right? And so, and just keeping that collaborative environment, just letting people just talk, bring up ideas, being respectful, and then coming to some agreement about who's going to be on the team and what we're going to do.
All right, so we are a little over time. We have 2 additional slides. So our next seminar is October 8th. Same time, same place. We have more of a social science aspect to the presentation, understanding and perhaps reducing hypothetical bias through collaborative research with Drs. Jacqueline Mito and Catherine Kling. And this talk will be recorded and posted to the INRC website under our videos tab, along with all of our archived previous seminars. And then I want to give one last thank you to our presenters. It was a great talk. Thank you very much.
Thanks, Damon. Have a good evening.